Good day to all and welcome to Raaonline. So now we are going to attend a series of microbiology lectures, and I am Dr. Priyadarshini, a professor of microbiology, and I had my undergraduate study at Kilpauk Medical College and my post-graduation from Madras Medical College. And I have about 15 years of experience in teaching undergraduate students and postgraduate students. So in general bacteriology, there are a lot of important topics to be covered as per the CBME as well as the old curriculum. So among them, the study of bacteriology becomes interesting only when we know the basics of the bacteria. Okay. So in that series, the first of these lectures will be morphology of bacteria. So what is morphology? You're going to study about the structure of the bacteria, the size, then the staining properties, and all that comes under the morphology. Okay. So before knowing the morphology of the bacteria, let us go to the learning objectives for this session. So at the end of this session, you will be able to know a few things. So the first one will be difference between prokaryotes and eukaryotes. Then what is the exact definition of bacteria? Then the structure of the bacteria as well as its organelles. So we will be studying the morphology of the bacteria as well as the organelles. Organelles are the cell structures which are present inside the bacteria. Then the different staining characteristics of the bacteria. So we will be seeing all this in detail. Okay. So the first thing is, right from your 9th standard and 10th standard, you students would have studied about the plant kingdom as well as the animal kingdom. Initially, some of the microorganisms were classified under the plant kingdom and some of them were classified under the animal kingdom. But now, they have been classified under a separate kingdom known as Protista. So members of this kingdom Protista are known as protists. And these protists have been divided into prokaryotes and eukaryotes. So you know the word karyo, okay? It means something to do with the nucleus. So here they are divided into pro and eukaryotes based on difference in the cell structure as well as their nuclear organization. So not only cell organization but also the nuclear organization, the biochemical properties and the functions. So karyo means, as I told you, something to do with the nucleus and the nuclear material, and pro means it is primitive. So these prokaryotes are usually primitive microorganisms, and eukaryotes, eu means good or true, and these means they are more advanced microorganisms. Okay. So a few examples of the prokaryotes are bacteria, also blue-green algae, and examples of eukaryotes are fungi and protozoa. So now we know what exactly the protista are. Based on this, we have to see the differences between pro and eukaryotes, and this can also be asked for you in your viva or as well as in your practicals or even in the theory. So difference between pro and eukaryotes is based on difference in structure of the nucleus, differences in the cytoplasm, as well as difference in the various chemicals that are present in the cell. So this nucleus and cytoplasm differences we will be discussing as we are studying the structure of the bacterial cell. I will also tell you the differences between the nucleus and cytoplasmic structure. And chemicals, you just have to know that sterols are chemicals which are present in fungi, that is they are the eukaryotes, and they are absent in the prokaryotes. And muramic acid is a chemical which is present in the prokaryotes. So this is a major difference in the chemical structure. Okay. Now we will go to the definition of bacteria. So before going on to the structure of bacteria, we need to know what are these bacteria. So they are unicellular microscopic organisms, and here one cell will perform all the essential functions of life, meaning growth of the cell metabolizes as well as the cell reproduces. Here one cell will perform all the structures. We all know that in our body, we have different organ systems. Each one is responsible for one function. So you have one system for kind of doing the digestion. You have an excretory system. Then we have a central nervous system and so on. Whereas here, they're all very primitive organisms. Only one cell is there, and that cell can do all these functions like growth, metabolism and reproduction. And one more important feature of bacteria is that they contain both DNA as well as RNA. DNA is deoxyribonucleic acid and RNA is a ribonucleic acid. This is to differentiate them from the viruses, which are another group of microorganisms, which will have either DNA or RNA but never both. So under morphology, the first thing, the point that we'll be seeing is the size of the bacteria. Okay. So the bacteria are very small organisms, as we already know. They are microscopic. Microscopic means you cannot see them with the naked or the unaided eye. Why you cannot see them? Because their size is very less. What is the size of the bacteria? It is in microns. And how much is a micron? We all know that 1 millimeter is 1,000th of a meter. Okay, so we know that a millimeter itself is so less, and a micron is 1,000th of a millimeter. So you can imagine how small the bacteria is. So you will not be able to see the bacteria. You know that bacteria are present all around you, but you are not able to see them because their size is so small. So the usual size is 0.2 to 1.5 microns in diameter, and the length is 3 to 5 microns in length. There are bacteria, usually the saprophytic bacteria, meaning those bacteria which are present in the environment and which do not cause any human disease, their length may even be greater than this 5 microns. This is the dimension which is mentioned for most of the pathogenic bacteria. Pathogenic means causing diseases either in humans or in animals. Okay, right. Then so we have come to the conclusion that when you want to study the bacterial morphology, you need the use of a microscope.